Alrighty, it is 2:15. We are back on schedule, I think. It's my great pleasure to introduce Dr. Peter White, who heads the biomedical genetics core at the Research Institute for Nationwide Children's Hospital, and today he'll be talking about population scale genomics algorithms that he's done. Following him will be Abi Naloor from Johns Hopkins University, talking about RNA-seq analysis. So two great talks about going to scale in genomics. As we know, genomics is becoming a problem, a big data problem. Without further ado, Dr. White.
Okay, I'll tell them if I need to go back. Hi, yes, good afternoon, everyone. As Angel said, my name is Peter White. I'm from Nationwide Children's Hospital, which is in Columbus, Ohio. And I'm going to be talking to you about some work that we did earlier this year as part of the Intel Head in the Clouds Challenge that David Everard mentioned in his keynote speech this morning. And so that'll be in the second half of the talk. I'll talk about that. But before I get into that, let me give you some background on genome sequencing and what's involved. So the Human Genome Project began in 1991, and it was an international effort to sequence and assemble the structure of the first human genome. It took these papers came out 10 years later in 2001. This was the release of the first draft of the human genome, but it wasn't actually completed until the final draft was in around 2006. So you're looking at about 15 years and a cost of $3 billion to sequence a single human genome. Now, this is what it would look like if you were to print out that genome. This is housed in the Wellcome Collection in London. It's a collection of over 110 volumes. Each volume is containing just pages and pages of A, C's, G's, and T's. And I'm actually headed to England with my family tomorrow, and my kids think they're going to get a trip to the Tower of London, but we're going to go see this instead. So today, we can sequence genomes a lot faster. And so this is sort of giving birth to a new era where we can actually sequence thousands of genomes. And this is because of a new type of sequencing technology that's called next-generation sequencing technology. Now, in my lab at Children, we have two of these next-generation sequencers. This little guy here is called a MiSeq instrument. It's great for sequencing bacterial or viral genomes or for doing smaller studies. But our main workhorse is this machine, the Illumina HiSeq 4000. This is capable of generating 5 billion sequencing reactions in parallel. That's about 1.2 terabytes of raw data coming off this machine every 3.5 days. But that amount of sequencing output allows us to sequence up to 12 human genomes every 3.5 days. So the focus of my research has been in an area of translational bioinformatics. How do we turn all of this data coming off of these machines into something meaningful in terms of molecular diagnostics and clinical treatments? So it would be nice if the data coming off these sequencers actually looked anything like this, but it doesn't, because the way next-generation sequencing works is the genome has to be chopped into very small pieces and then sequenced in parallel. So it's a bit akin to taking this entire collection and running it through a paper shredder. This is what the output actually looks like. So the analysis process has to reassemble all of those volumes and get them looking back to something readable. And then what we're looking for is that one page in one of those volumes where a letter has changed. In this case, we have a C that's changed to a T, and often this is what could be responsible for a genetic disease. So that's what we're doing in the analysis, going from that shredded output to finding a pathogenic, what we call a pathogenic genetic variant. So when we first started doing this, the problem that we ran into very quickly was this instrument in a high-speed mode can sequence a human genome in as little as 24 hours or around 2 days. And it was taking us up to 2 weeks to do the data analysis. And so our solution to this problem was to come up with a new approach to parallelize the analysis. And we called that solution Churchill. And what it essentially does is it goes through each of the steps that are required in the analysis process and very efficiently performs the parallelization. And so I'm not going to go into any of the details of specifically how that works, But we published this earlier this year in Genome Biology. And so if you search for Churchill and maybe my name in Genome Biology, it should be a top hit in Google. But so when you look at strategies for doing this workflow, there's a couple of ways that you can paralyze it. What others have done is they've used a natural way to do the parallelization, and that's by parallelizing the analysis by chromosome. So the genome's comprised of the 22 autosomes and the 2 sex chromosomes. So here you could see you could parallelize this analysis over the 24 chromosomes. There's two problems with this approach. First of all, it's imbalanced. The chromosomes differ in size. Chromosome 21 is the smallest at around 50 megabases, but chromosome 1 is the largest at 250 megabases. So any analysis that's parallelized by chromosome will be dependent on the speed it takes to complete the largest chromosome. The second thing is you're limited to 24 parallel processes. There's only 24 chromosomes that you can parallelize the analysis over. And so what we did with Churchill is came up with something that we call a balanced subchromosomal region parallelization strategy. And that's why we called it Churchill. That was just too big of a mouthful to say that. But essentially what we're able to do is to take each chromosome and break it into smaller subregions. And so you can see the result is that we can then balance the analysis process. Some of the benefits of this is that the Churchill algorithm is very highly efficient in terms of the compute resources that it utilizes. So what I'm showing you here is CPU utilization, and then the time that it takes to complete analysis of a whole human genome. And when we compared this to two alternatives to Churchill, a pipeline called HugeSeq from Stanford and a pipeline called GATK-Q from the Broad Institute, you can see that Churchill was a lot more efficient and was able to complete the analysis in the shortest amount of time. And this is on a single machine. What you can see here is this drop in utilization closely matches the chromosomal size, and this because HugeSeq is using a chromosomal parallelization strategy. So with Churchill, we're able to scale beyond a single computer or a single instance, and to use multiple instances and divide the genome into smaller and smaller regions, allowing us to speed up the analysis process. So what I'm showing you here is this is speed up, and this is the number of CPUs. So the yellow and the green are GATK-Q and HugeSeq, and they're really limited to that 24 parallel processes. But with Churchill, we closely track with a computational prediction of how much you can speed this process up. It was predicted by Amdahl's law, so Churchill's the red line here. And then as a result of this, we're able to reduce the analysis time substantially. And in this case, by the time we're out to, this was 16 instances on EC2, so that's equivalent to 768 CPUs. That allowed us to do the analysis in about 99 minutes. And right now, we're running at around 77 minutes to do the analysis. So we're trying to get that down to under an hour. But I work in a children's hospital where speed can be critical with newborns that have suspected genetic disease, but also accuracy is very important for us. We don't want to sacrifice quality for speed. And what we were able to do is using a standard that came out from NIST last year, something that's called the Genomes in a Bottle standard. This is a standard genome that has validated variant calls. We're able to compare the output of Churchill to the other pipelines. And what we found is that across the board, Churchill had the highest number of validated SNPs, the lowest error rates and highest accuracy. And then in terms of diagnostic effectiveness, we use a measure that's called the Uden index, which is the sum of sensitivity and specificity minus 1. We saw that we had the highest overall diagnostic effectiveness. The other part of this is the Churchill algorithm is 100% reproducible and deterministic. And what I mean by deterministic is that regardless of the scale of parallelization, regardless if I run it on my cluster or you run it on your cluster or you run it in the cloud, you'll always get the same results back. And it's the only pipeline out there that has those features. So how does this tie into population scale genomics? Well, in June last year, Children's launched in partnership with JHC Technology, Nationwide Children's launched a new company called Genome Next, which is a genomic analysis software as a service company that provides a cloud platform for running genomic analysis, and Churchill was one of their flagship pipelines. And I was at this meeting with James Hermes, the CEO of Genomics, when we heard Intel announce the Head in the Clouds challenge. And I said, at the time we sat with each other and both had the idea, what would happen could we take Churchill and run this on a really large data set? So what we proposed was that we would analyze the Thousand Genomes data set, which is one of the largest publicly available genomic data sets that are available. And so we submitted an application, and we said that we'd like to be able to scale Churchill using AWS and see if we could perform the analysis of that entire data set in under a week. So the Thousand Genomes was a project that began in 2008 and was conducted by an international consortium of over 400 scientists and was designed to capture really and create a catalogue of human genetic variation from around the globe. And it's been done in a number of phases, and as the cost of sequencing has dropped, they were able to actually increase from 1,000 genomes to a final sample set of 2,504 genomes. And this was from 26 populations around the globe. Now, they completed, I actually need to go back one, sorry. Can we go back? Yep. They actually completed phase 3 of the analysis in April 2013, or phase 3 of the sequencing. All of the genomes had been completed in terms of sequencing. And it wasn't until 18 months later that the analysis of this was completed. And in September last year, they released the final results. And I think a publication for this work is coming out later this year. So in terms of being able to analyze a population-scale data set, one of the challenges that we had to overcome was data upload. The final 1 kg data set consists of 2,504 exome analysis and whole genome analysis for each individual. So they're just two different strategies to do sequencing. The total size of that data set is 70 terabytes. So the challenge is, how do you transfer that amount of data quickly in order to be able to perform the analysis? And the answer is you can't transfer it easily, so you take your analysis to the data. And so that's what we did with Genome Next. We were able to because all of the data for this project has been stored and made available in the S3 bucket, we're able to co-locate our analysis platform in the same region where the data was stored, and that's in the US East region. So this is an example of how the platform that was developed to run the analysis, this is a screenshot. What we wanted to do is to be able to try to fully automate as much of the process as possible. So it started here with us importing a sample index. And then essentially what we had to do is provide an Excel sheet that contained each of the sample names and then the location in the S3 bucket where that FASTQ data was stored. And then when this Excel sheet was uploaded, all of the analysis was kicked off automatically. So the second challenge we had to overcome was in terms of scale. Analyzing, what I talked about earlier, I was showing you analysis of a single genome. Now we're talking about analyzing over 5,000 samples. And so this was the overview of the infrastructure that we put together. What we're starting with here, this is the Genome Next portal that would import the data from the S3 bucket and then performing the analysis and then delivering the results back into S3. And to go into the details of that, a bit more detail, you're starting here with, so this was us in our lab in Columbus submitting the request, all of the data, so we submitted our Excel sheet that contained the location of all the samples in S3. Then via a load balancer, that request goes to the Genome Next portal. What the portal uses is 3 different queue services. First of all, the analysis request service up here sends a message to what we call the Churchill listener, and this actually then deploys the infrastructure that's needed to perform the analysis for a given sample. So in this case, depending on the size of the input, if it's an exome or a whole genome, you could anywhere be from a single instance up to 16 instances per sample. The analysis then begins, all the infrastructure is launched, the analysis begins, and messages are sent back that report on the status of the analysis. And then once it's completed, the final results are delivered, the files are copied back to S3, and then the user is able to download the results back through the web browser. At the same time, all that infrastructure that you deployed is automatically taken down. Now, we did this using anywhere up to 1,000 instances over the course of the 7 days. And this was a schematic that we put together to kind of summarize the results. Essentially, we were able to take all 2,500 individuals, 5,000 samples, 85 trillion base pairs of data, totaling 70 terabytes. In input files, it was about 148,000 different input files, and then anywhere up to 1,000 instances, completing analysis in the 7 days. Now, in terms of what the results look like, because this data set had been analyzed by the Thousand Genomes Consortium, we were then able to compare our results that we got back to that original data set. And what we found in terms of the different types of genetic variants that we identified, we looked for single nucleotide polymorphisms and some of them are called indels. These are small deletions and insertions in the genome. For the SNP calls, we had a high degree of overlap with the calls that Churchill called and those that were identified by the Thousand Genomes. For the indel calls, we actually discovered just over 5 million indels that hadn't been reported by the Thousand Genomes. And we think this was because we used a more sensitive algorithm for doing the indel call, a more sensitive algorithm for doing the indel calling. And when we look at some of the validation rates of this data, we are actually seeing a high level of validation of these indel calls. But in terms of the overall variant frequencies, when you study a population genetics, you want to say, how common is this variant in my given population? If we observed a frequency of 20% in our analysis, the Thousand Genomes also reported a 20% frequency. So we had an extremely tight correlation for the overall population frequencies for the variants that we called in common. So with that, I'll finish up. I want to thank the support that we had from Genome Next in performing this, and particularly thank Amazon and Intel for funding the project and for allowing us to perform the Thousand Genomes analysis. Nationwide Insurance has been very supportive of our genomics program at NCH. And then this is my team. Most of the work I've described here has been done by Ben Kelly, who was the lead developer on Churchill, and James Fitch, who did most of the Thousand Genomes analysis. And with that, I'll stop, and I'm going to take questions after Abby's talk. So I'll hand over to Abby.
Okay. So hi, I'm Abhi Nalor. I'm a postdoc at Johns Hopkins University in computational genomics, working between the labs of Ben Langmead, who's in the audience here, and Jeff Leake. We focus on analyzing RNA sequencing data, and we try to do it at scale with Amazon Elastic MapReduce. And I first got into Amazon Web Services when I started this postdoc almost 2 years ago now. And now I'm an Amazon Web Services junkie. We actually have a couple of clusters running right now analyzing sequence read archive data. That there are 2 such clusters, each span about 2,000 processing cores, and that's the size of 2 clusters we would use at Hopkins. So we don't do it though because we're drunk with computational power. We do it for the science. I'm not sure what I'm supposed to press here. Play? The green button? Oh, okay. Okay, we do it for the science. And when we do computational genomics, we're interested in the aspect of molecular biology that is the flow of genetic information within living things, like you and me and others. So this is captured, a good first approximation to this is captured by the statement made by Francis Crick, this Crick central dogma, that is roughly DNA is transcribed into RNA, which is then translated into protein. I said we focus on RNA, but you might then say, well, why should you study RNA when isn't all the information encoded in the DNA? And no, that's not true at all, because DNA, I mean, that's kind of like saying you've listened to a Beatles record by looking at its grooves. You gotta play the record. So what we do is we allow the cellular machinery to unpack and interpret the compressed information stored in DNA and gain some insight into what these sequences of A's, T's, C's, and G's mean. Okay? Okay, so RNA is important. How do we find out about it? Well, you sequence it. And roughly what happens in a sequencing experiment in broad strokes is you extract RNA, you isolate and purify it, you fragment it, so it's about, maybe a few hundred base pairs long, segments a few hundred base pairs long. Then you convert it to complementary DNA, ligate on some adapters, you obtain short sequencing reads, and we deal with sequencing reads that are maybe between 50 and 300 base pairs long. Software you develop to analyze this data does that. That's what we do. So we created the software tool called RailRNA to analyze lots and lots of these sequencing samples. And why might you be interested in lots and lots of sequencing samples? Well, it just turns out that a lot of investigators are now analyzing a lot of sequencing samples. And you can see this if you look at these public archives of sequencing data. Let's see. So, if you do any kind of publicly funded research, if you're publishing papers, you're going to release the data you used, and you generally do this by putting it on a public archive. One of them is the Sequence Read Archive. Another is this European Nucleotide Archive. And they have these great plots on their site. You can see there are about 1 × 10⁻13 reads on the European Nucleotide Archive now. And I think that spans between 1 million and 10 million samples. And another interesting figure is the one on the right, which is the read doubling time. And that figure is essentially telling us, well, whenever you're doing something at scale, it's great to say that the data you're working with is growing in size exponentially. And that would be true if the plot on the right were a flat line or approached a flat line as the number of years or the year increase. But what it's actually saying is that in about 2 years, we will have doubled the amount of data on this archive that's been accumulated up to that point. And that spans seems like 7 years, which is pretty impressive. So why? Why are we getting more data on these archives? And it's because the cost of sequencing is dropping and the experiments are getting larger and larger. Genomics initiatives they're accumulating these large RNA-seq data sets. And probably one of the pioneers in this was the ENCODE study that now, it seems like a paltry number, but they did 100 RNA sequencing samples. But they actually did a study that spanned a lot of next-generation sequencing technologies. There's the Giovanni study, which spanned 465 lymphoblastoid cell line samples. Depression genes and networks, 965. This is divided into 2 groups. Half of them have major depressive disorder, the other half didn't. TCGA, over 2,000 cancer samples. And recently, the GTEx consortium released about 10,000 RNA sequencing samples, and there are 175 individuals. These are difficult to obtain tissues, maybe 43 difficult to obtain tissues across these 175 individuals. And they still haven't been analyzed completely. So you might ask yourself, okay, so all these other people are caring about a lot of samples. Why should I care about their data? Why might I want to reanalyze their data? Because if you're making a tool to analyze a lot of samples, well, why not analyze samples that already exist? You're repurposing rich, hard-to-obtain data. As I said, GTEx, these are deceased individuals, these are tissues that come from them. Where are you going to get this data? Are you an investigator with limited resources going to be able to get this yourself? Probably not, but someone else, several groups, a consortium's scale of groups has done this already. Another reason is just no 2 analyses are commensurate. Okay, you might do an analysis one way, another guy might do an analysis another way, and you might disagree with existing analyses. Something as simple as how you normalize the coverage of your sample across a genome can significantly affect your results, and you might have to redo someone else's analysis, especially if you're trying to analyze a lot of data from different experiments. To make them commensurate, you'll have to rerun your pipeline on that data. Another great reason is to ensure the reproducibility of someone else's results. In science, you need to verify that someone else's results are, someone else obtained results in perhaps the right way, or verify the effectiveness of the statistical methods used by other investigators. Or you might add power to your experiment. You might have done a study with lymphoblastoid cell lines, and you might augment the power of that experiment with experiments from the Geovatis study. So there's some incentive to reanalyze publicly available data. But why should you do it now in the cloud? There are two one-word answers to this question that I see. One of them is elasticity, and the other is reproducibility. In elasticity works in two directions. In one direction, it's elasticity in compute. So if you're using your institutional cluster, you're going to submit a job to some queue. And it's going to sit there in the queue, and it could be sitting there in the queue for a night, right? And I mean, you're hoping that there will be compute available for you to do your job. You're just praying that it'll start. Well, I mean, if you use a service like Amazon Web Services, you'll submit your job. And if we use Elastic MapReduce, and we typically find that within 10 to 20 minutes, our job starts. And it'll start with as many processing nodes as we request. So it's not like we have to wait patiently for enough nodes to become free on an institutional cluster. So there's elasticity in compute, but there's also elasticity in storage. With S3 pretty much means you have infinite storage. If you're analyzing a lot of sequencing samples, for example, the Geovatis study, on a per sample basis, it's about 2 gigabytes per sample. But the intermediate data our pipeline accumulates takes up about 10 times that amount. So let's say we're analyzing 10,000 samples. That's going to be about 200 terabytes, which your institutional cluster might not have available. So using a cloud computing service like Amazon Web Services frees you from concerns about having as much compute as you want and running out of space. Another great reason to use a cloud computing service to do your analyses is you're not going to find a more reproducible system unless you use another investigator's hardware and software in exactly the same configuration. The next best thing is for you to use the same hardware type, the same instance types, the same size of the cluster, and the same software version, the software configuration, which the software we developed helps you do. All right, maybe I messed up the title of that slide. But the point of this slide is also to say that you might use any cloud service provider, but a problem you run into when you're working with distributed systems is consistency. And consistency is when you're working with cloud storage like S3, you might, there's like a transaction. You might say, I write a file to this cloud service, to this cloud storage system, and then I try to read after it, and those two operations might not be consistent. I might not get back exactly what I wrote. And so there's some delay. This is an eventual consistency. So now all Amazon regions have so-called read after write consistency, but you might actually have deleted a file, and then you might list a directory on S3, and after you've deleted the file, that file might still be present. You might update a file and then try to retrieve it, and you might get the original version of the file. And one way Amazon addresses this is through EMRFS, which we use in our software to ensure that when we write to S3, we get fully consistent results. And I don't think any other cloud service provider has this feature. So that's advantage EMR. All right, so back to the problem we're trying to solve. When you have these RNA sequencing reads, you want to find out where they originate in the genome. And so you align it to the genome. And sometimes you're lucky, and a read correctly aligns to the reference genome end to end. And this is an example of such an alignment. But sometimes you're not, because RNA has segments of DNA missing from it called introns. And these are segments from the genome that aren't expressed. So when these introns are overlapped, okay, there's going to be a piece of the genome missing from the sequence read. And you'll have to align that read in pieces. You'll divide the read up into short overlapping segments. This is what we do, called readlets, okay? And then we'll align the readlets to the genome to infer the presence of introns. So you'll see on one side here of, I don't know, is there? Oh, great. You'll see on one side of this intron, you'll have this short segment of the read, it's 25 base pairs long, and aligns to one side of this intron. This side of the read, the right side of the read, aligns to the other side of the intron, okay? And we can easily infer the presence of this intron. But sometimes reads won't overlap the intron right in the middle of a read, right in the middle of that read right here. Could overlap the intron towards the end of a read. So what we'll have to do is accumulate a list of introns, okay, from that sample or across samples. Okay, and then knowing the presence of that intron, we create segments, we take segments of the genome around the intron and concatenate them and then realign reads to infer the presence of the intron. So the software we created is called RailRNA, and with just 1 to 2 commands, you get alignments just like the one illustrated on the previous slide, introns and their coverage, indels, insertions and deletions in these reads, and just coverage information, which parts of the genome are covered by how many reads. And all we do is you have one file, and you list all your FASTQs. They could be they could be on the web, they could be on an FTP server, they could be local. You specify the assembly, and this one's HG19, human reference. It's not. Then we specify the number of instances we want to use, the number of computers we want to use in the cloud, and an output directory, which is an S3, which is a directory in an S3 bucket. And you can download it at rail.bio. Great. So how does this software work? How do we actually achieve our ends? You want to scale. You want software that gets faster when you have more workers in a cluster, and you also want it to work on a lot of data. You don't want it to break when you suddenly reach 1,000 samples or 5,000 samples. So the approach we took was the MapReduce programming model. And you can understand MapReduce as you take a computer cluster and you divide it into a set of workers, and you take a computing task or a set of computing tasks, and you arrange them so they're a sequence of alternating aggregation and computation steps. In these aggregation steps, the data are grouped and sorted, and the computation steps, each worker operates on some subset of the data. To illustrate how this works in Rail, I'll actually describe how Rail exploits this MapReduce programming model to reduce redundancy in analysis. And the idea is, roughly, that you're going to have lots of reads across these samples. And there's going to be a lot of duplication across these reads, a lot of duplicate sequences. You don't really need to align these sequences more than once when you're just looking for introns. Similarly, when you divide the read up into these short readlet sequences, there's a lot of duplication across these readlets. And you don't have to align identical readlets more than once. So what we do is, this is just a part of the pipeline. We aggregate. In this aggregate step, we group reads by their unique sequence. And then we segment these unique sequences, group the unique readlet sequences, and align the unique readlet sequences. So this reduces the alignment burden so significantly. Then we aggregate again after aligning these unique readlet sequences, so we can associate the readlets back with unique read sequences, the read sequences to which they belonged. This is another aggregation step. And then we search for introns within each unique read sequence. All right? And this goes, this is an interesting story because here's part of the story is how we get improved throughput with RailRNA when we increase the number of samples we analyze. So naively, you might say, okay, I analyze 10 samples on a fixed cluster, computer cluster of fixed size, say 100 computers, and I analyze 20 samples on a cluster of a fixed size. I wouldn't expect that I analyze more samples per hour when I analyze more samples, right? So naively, I'd expect that it's constant. My throughput is constant as I increase the number of samples I analyze. But RailRNA has improved throughput the more samples you analyze, simply because it's reducing redundancy in its analysis. This is illustrated here on this plot, where we analyzed increasing numbers of G/Vaudis samples on the same cluster of 40 C3.2x large computers or instances. That's 8 processing cores, so it's 320 processing cores in total. And you can see that as we increase the number of samples, here, 2, 3, we get improved throughput. And improved throughput actually means that how much it costs to analyze a sample goes down the more samples you analyze. We use spot instances, and that's why these prices might seem lower. And it cost us, we bid, I think, 11 cents. So we paid up to, because there's an Amazon, there's a surcharge, we paid around 25 cents per instance per hour. So can we go back a few slides? Back. Yes, okay, that's perfect. Okay, so that's the scalability story with respect to number of samples. But of course, there's also speed up. In practice, so ideally, you might say, okay, I use twice as many computers, I do things twice as fast, but in practice, there are network bottlenecks, there are I/O issues. So we scale okay, but it costs more the more computers you use to analyze your samples. So what we did was we compensated for this effect with this reduced redundancy analysis. Analyze more samples on more computers, and these two effects cancel each other out in some sense, and you get pretty good scale. So that's what we did. One of the milestones, so we analyzed all of G/Vaudis in just 12 hours on 1920 processing cores. It cost us 69 cents per sample, and spliced alignment is a pretty hard task. To get all those results, coverage information, indels, alignments in 12 hours, we feel it's an achievement. And next up is for us to analyze all of human Illumina RNA-seq on SRA. And as I was just describing, we're doing this. We're in the process of doing this right now. We have clusters doing this right now on EMR, and we're very excited about how we're going to use this tool in the future. And we hope you try to use it too, if you're interested in doing this kind of work. So rail.bio. Thanks a lot.